Hello, today I want to show you how to do nickel affinity purification. It is a common way to purify proteins from your cells, which have a lot of different types of proteins. And it's very simple. You need 3 buffers, one a resuspension buffer that doesn't have any imidazole, one a wash buffer that has 20 millimolar imidazole, and an elution buffer that has about 300 millimolar imidazole, which is the highest amount of imidazole from the 3 buffers. So after adding 5 mils of nickel resin, as you can see it's 5 mils, and letting the ethanol that was in it pass through, we can add the resuspension buffer, and this will equilibrate the resin. And I usually add about 50 mils in total, so about 2 of those column volumes. And meanwhile, I will spin down my lysate with about 8,000 RPM for 30 minutes at 4 degrees. This is because you want to spin down all the residual junk that's in the lysate, so it won't block your resin. And each time you use the resin, small amounts of it will get in and it will become more slow. But it's crucial to spin it down before adding it to the resin. So after equilibrating it, you add your lysate, which contains your protein of interest, as well as all the other proteins that are in the cell. But your protein is supposed to have a his tag, which was encoded in its gene, and then that's how it got expressed. And this is what will allow it to bind the nickel and chelate it. But after that, you will add the wash, and that will get rid of other proteins, but your protein will still be bound to it. And then finally, you add the elution, which has the highest amount of imidazole, and imidazole will instead bind nickel, and then the histidine tag will let go of the resin, and that's how you elute your protein. You can do this purification either in denaturing or non-denaturing conditions. For me, it was denaturing with 6 molar guanidine, and then I had to refold it. However, if you're doing in a non-denaturing condition, do all the steps in the cold room. Thank you so much for watching, and if you have any questions or suggestions, leave a comment below.